There are three major ways to get DNA from one organism to another organism in the bacterial world. One is by transduction. A virus would infect one cell, pick up some of the DNA from that, and then move it over to another cell and inject it into the other one, and that can then become part of the genome of the second organism. Another one is conjugation. Two cells come close to each other and form a pore between them and pass DNA from one cell to the other. And then the third way is called transformation. That's where bacterium sitting in liquid sees naked DNA floating by from a cell that may have ruptured and brings that DNA, pulls it in through its membrane. All of those are powerful modes of evolution because it's not just that it gets to try out lots of other genes, it also gets to try out genes that it runs into. This is the organism.
Yeah. Right. Yeah, it's got that green.
One of the things with USA 300 is that it makes more of that alpha toxin. That may be one of the reasons why USA 300 is more virulent. The first thing we did was look at the genome. The USA 300 genome had been sequenced by two different groups, therefore served as a great way to get to know all of the genetic material in this one organism. By comparing that to other known Staph aureus genomes that were out there, we could immediately see basic differences between that genome and other genomes. While USA 300 has this big, long piece of DNA here, it's just completely absent in the other strain. Some of those differences were on the order of whole genes, so there were whole new genes that nobody had ever seen in Staph aureus before in this genome. But there was a gene that sort of popped out of our analyses when we were looking very closely, and this was the SPG gene. SPG is something that takes spermidine and chemically changes it. Spermidine is something that humans make in their skin. It is made when you have a cut when you're healing. And we know that you can kill Staph aureus with spermidine. But we found that USA 300 is completely resistant to the killing power of spermidine. Nobody had really thought about spermidine and its toxicity to staph in a very long time. So it led us to ask questions about what the role of spermidine might be in Staph aureus infection.